Genes are extremely small, only making up a small percentage of your DNA. But their impact on human evolution and the world is extremely big. And you'd be surprised that the entire field of genetics started from something so small as just one man and a pea. And equally surprising, you did too. Not from a pea, but from a small beginning. But how did something as big as genetics, or you, start from something so small? Well, to answer that, I think it's best if we start small too. So come along with me to the beginning of genetics to see exactly how big genes had small beginnings. Artwork for the human being was created by mx.monotone. Check them out on Instagram for more amazing works. This man is Gregor Mendel. He was an Austrian monk in the 1860s, and one day he began to breed pea plants. He noticed that breeding pea plants with two factors, or variants of a trait, resulted in the next generation all having the same variant. But when Mendel bred those new pea plants together, only 3/4 of the next generation had that variant, and the other 1/4 had the other. This worked for 7 different traits in every single pea plant. Now, Mendel's experiments were only done on traits determined by a single gene, and I'll get into why that's important later. But his simple experiments on pea plants still had a huge impact on both science and the world. He discovered the first type of genetic inheritance, now known as Mendelian inheritance, which helped him develop 2 very important laws of genetics. First, the law of equal segregation, which says that when a sperm and egg cell form, the allele they receive is completely 50-50. And second, the law of independent assortment, which says also when sperm and egg cells form, the assortment of a trait or gene is independent from another trait or gene. With this, Mendel founded the entire field of modern genetics, which gave rise to even more big discoveries, one of which being who you genetically are, which is pretty big if you ask me. However, Mendel's research wasn't discovered until 30 years after he published it, around the late 19th century, which was when the science of human evolution, paleoanthropology, was having its own small beginning. Genetics wasn't a part of human evolution for much of its beginning, and even in its early days, little was understood about them both, leaving a small amount of discoveries. That is, until the 1980s, when some researchers decided to use both genetics and human evolution to answer a very important question. Where did humans evolve? Normally, researchers use nuclear DNA, located in the nucleus, because it gives more quality information. But they decided to use mitochondrial DNA instead, located in the mitochondria. They did this because overall, there is more mitochondrial DNA than nuclear DNA in humans. Mitochondrial DNA is only inherited from the mother, which makes it good for relating people to one another. And mitochondrial DNA mutates faster than nuclear DNA, making it different among all people and reflect the full human diversity. Then, when they looked at all the mitochondrial DNA, they saw as you went out of Africa, the less diverse or younger the mitochondrial DNA was. From this, they formed the out of Africa hypothesis and discovered that all humans evolved in Africa around 200 to 300,000 years ago. So by using DNA in a non-standard way, especially when genetics was not well understood, they were able to make a big discovery about our own species' small beginning. By the 21st century, we knew that DNA was made up of genes, which are the basic unit of heredity that codes for traits. These are what you express from your biological parents, half of them from your mother, and half of them from your father, to make the whole of you. However, we didn't know how much of our DNA was made of genes. This was discovered from the Human Genome Project, which was a big international project to sequence the entire human genome, all of its 3.2 billion base pairs, which are the fundamental unit of DNA. Some researchers thought that genes made up 10% of our DNA, but actually, genes only make up around 1 to 2%. So at least 98% of our genome doesn't code for traits. This is called non-coding DNA, which instead helps regulate and control gene expression. Now you may be asking yourself, if genes make up such a small percentage of my DNA, then how can they have such a big impact on my life? Well, genetics is fascinating. One gene can actually code for multiple traits, which is known as a pleiotropic gene. Simple genes that code for size, shape, and function are examples of this. But also, multiple genes can code for only one trait. This is known as a polygenic trait. Complex traits such as height, weight, and skin color are examples of this too. So going back to Mendel, this is why his experiments on traits determined by a single gene, although revolutionary, were small beginnings in comparison to the discoveries of how we know DNA works today. And today, we even know how ancient DNA works, thanks to Svante Pääbo, a Swedish paleogeneticist and 2022 Nobel laureate, for his research getting ancient DNA from Neanderthals. And if you're as fascinated about ancient DNA and paleoproteomics as much as I am, and want to learn more about his small beginnings and big discoveries, I made a whole episode about it, right here. Genetics started from something so small, just one man growing pea plants, but has evolved into a major field of human evolution and science in general, creating some of life's biggest discoveries. And the same goes for you too. Big self-discoveries often start from small beginnings. And hopefully, by understanding the small beginnings of genetics and how it led to its biggest discoveries, you can better understand this in yourself too, and finally discover that big things have small beginnings.